Okay, this is the last part of this lecture, part 5. And we talked about the tyrosine kinases, different types, different kinds. Now let's talk about the multi-tyrosine receptor kinase inhibitors. Okay, we said earlier in the previous part that people are going toward selective inhibitors. So what's the reason for using multi-receptor kinase inhibitors? All right. These inhibitors designed to be selective against a range of tyrosine receptor kinases implicated in tumors. So it's multi-receptor inhibitors, but it's just inhibitors for those that are implicated in tumors. The reason why we are using these because drug resistance unlikely to occur for all kinase targets. So if a drug can inhibit 3 or 4 targets, okay, even if the cancer is able to develop resistance, okay, for one of those targets, maybe 2 targets, okay, but the cancer is not gonna develop resistance for all of them. Equivalent of combination therapy, okay? This is equivalent of combination therapy. Instead of using 3 medications and each medication will attack different targets, maybe you can use one agent that can attack those 3 different targets. It's a term that is referred to as polypharmacology, and I think you might learn about that in your pharmacology class, maybe or maybe not. It's also sometimes called promiscuous drugs, and I think you know what promiscuous term means. Promising agents against tumors that are driven by several abnormalities. So these agents, if the tumor is driven by several abnormalities means several or different abnormal targets, okay? Like an enzyme that stays active all the time. It doesn't become inactive. It's always active. That's abnormal, okay? So if the cancer has several abnormalities, these agents are promising. All right, examples. Examples on multi-tyrosine receptor kinase inhibitors like the sorafenib, and you see the structure over there, and the sunitinib with its structure. All right, sorafenib approved in 2006 for treatment of liver, kidney, and thyroid cancers. You see different types or different kinds of cancers. Also, sorafenib inhibits VEGFR, PDGFR, c-KIT, and RET. While sunitinib approved in 2006 also for treatment of gastrointestinal stromal tumors. See, tumors, not tumor, tumors. And advanced renal cell carcinoma. All right. Sunitinib also approved in 2011 for treatment of advanced pancreatic neuroendocrine tumors. So gastrointestinal stromal tumors, advanced renal cell carcinoma, and advanced pancreatic neuroendocrine tumors. Different kinds of tumors. Sunitinib inhibits VEGFR1, VEGFR2, PDGFR-B, c-KIT, and FLT3 kinases. Different types, different kinds of kinases. All right, other examples like the pazopanib with its structure here, and the vetilanib. All right. Vetilanib is still undergoing clinical trials nowadays. It inhibits VEGFR, PDGFR, beta, and c-KIT. Pazopanib, on the other hand, approved in 2009 for treatment of renal cell carcinoma, and in 2012 it was approved for the treatment of soft tissue sarcoma. Pazopanib inhibits VEGFR1, VEGFR2, VEGFR3, PDGFR, and c-KIT. Okay, we saw the examples. So now let's go to something that I think it's interesting to you, or it should be. If it is not interesting, it should be interesting to you, which is the design of sorafenib, the design of the drugs, how scientists design those agents. Okay, so lead compound found by high throughput screening, approximately 200,000 compounds were tested. Okay, they tested against recombinant RAF1 kinase. Sometimes they call it CRAF. Okay, this is the lead compound for sorafenib. As you can see, it's a urea derivative. It has this urea nucleus in it. Okay. In the lead compound, the R group is hydrogen. Okay. The IC50 for this compound is 17 micromolar. Okay. It is good. It has good activity, but it is not that good. Okay. You need to improve that activity. So let's see what scientists did. Well, first they tried what's called traditional medicinal chemistry, or if, as we call it here in Iraq, pharmaceutical chemistry. We don't call it medicinal chemistry, we call it pharmaceutical chemistry. But anyway, it's the same, or at least we use it as an interchangeable terms. Okay, so as I said in the previous slide, the IC50 for the lead compound is 17 micromolar. They tried to they used traditional medicinal chemistry, which means you change one group at a time. Okay, so you change the R group and you keep everything else. Maybe you change this group, but you keep everything else the same. And you test for the effect. Okay, and based on the results, you make your structure activity relationship. They found that if you have methyl group here at the para position, just a small methyl group, the IC50 improved 10 times. Here it is 17 micromolar, but for compound number 2, the IC50 is 1.7 micromolar. Okay. On the other hand, if you use a phenoxy group, which is phenol and oxygen, at this position, you will get poor activity. They draw a conclusion that big groups are not good for activity. Okay, you only need small groups like methyl. If you use big group like phenol, here phenoxy, which is phenol and oxygen, you get poor activity. All right. As we said, the methyl substituent in structure 2 is optimum for activity, 10-fold increase in activity. The phenoxy group is bad for activity, as we saw earlier. All right. They tried, now, as we said, they tried the traditional medicinal chemistry, but they couldn't get any improvement further than the 1.7 micromolar? Well, you need something in the nanomolar range. Okay, so they tried changing the ring here. Okay, when they used the isoxazole ring, this ring, they with the methyl group CH3 here, they got poor activity. So they said, well, that means the methyl, and because they draw the conclusion that the methyl group here is good for activity, they said, well, the isoxazole ring is not good for activity. It is bad for activity. It's just like the phenoxy group. Okay. So as I said, variation of rings also carried out systematically. Isoxazole ring is not good for activity. The conventional medicinal chemistry strategies fail to achieve further improvements because the best analog they got was compound number 2, the one that you saw in the previous slide. All right, then they decided to go and follow another approach. Okay, what's the other approach that they followed? They use parallel synthesis. Okay, they synthesized 1,000 compound with different variation and they tested them. Okay, something that you will learn about next year in combinatorial chemistry. The parallel synthesis, as I said, they synthesized 1,000 analogs, okay, with all possible combinations of rings and substituents. Rings here and substituent there. Structure 4, the one that you can see, has slightly increased activities. Well, slightly increased activity if you compare it to structure 2. Because if you remember, the IC50 for structure 2 was 1.7. Now the IC50 for structure 4 is 1.1 micromolar, which is slight improvement. But look at structure 4. It has the phenoxy substituent, which they said it is bad for activity, and it has the isoxazole ring. Well, how come 2 bad substituents give better activity? Well, the isoxazole ring and the phenoxy substituent are good for activity when combined in the same structure. Something that you know as synergistic effect. Okay, structure 4 now is taken as the new lead compound. Okay, instead of relying on the structure number 1 as the lead compound and keep modifying this one, we're not gonna modify this one, but we're gonna modify this structure from now on. Okay, so this is the new lead compound. Okay, this is the old lead compound. This is the new lead. Now, here the phenoxy substituent, the X is CH, right? Because this is a phenyl group, and since this is a phenyl group, this X would be carbon and the hydrogen, right? When they change it with a pyridine ring, okay, where the X is nitrogen, they got even better activity. The IC50 became 0.23 micromolar instead of 1.1 micromolar. So we call that ring variation strategy. And you see, now you are learning about different drug design and development strategies in each step. Okay, as we said, they got 5-fold increase in activity, also increase in aqueous solubility, because it's a pyridine that has a nitrogen which would be able to form bonds with water or ionizable salts, and improve in calculated log P. The calculated log P is important for drug absorption through membranes, like cell membrane. Okay. From structure number 5, by adding this group, they got the sorafenib. So in sorafenib, you have a substituent variation, well, a ring variation. Instead of the isoxazole ring, they have the phenyl ring. Instead of hydrogens here, you have chlorine and you have the trifluoromethyl, so which is substituent variation, and another substituent variation at the end of the molecule. That gives you sorafenib with an IC50 of just 12 nanomolar. So they go from 17 micromolar to 12 nanomolar, approximately 1,000-fold increase in activity. Okay. So what are the binding interactions for sorafenib? Well, here you see the structure for sorafenib, and as we mentioned earlier, the sorafenib has a urea nucleus in it. So this urea is capable of forming the required hydrogen bonding, because the amine act as a hydrogen bond donor and the carbonyl oxygen act as a hydrogen bond acceptor. You can compare that to the amide group in imatinib. It do the same job. The urea functional group act as a binding anchor. As I said, compare that to imatinib. The hydrogen bonds are formed to catalytic aspartate and glutamate. And we mentioned earlier that if you want your compound to be active, it has to have those hydrogen bonds. The binding orientates the molecule. It positions each half into 2 selectivity regions. It splits the molecules into halves. All right. That's what we have about the multi-tyrosine kinase inhibitors. Now let's move on to kinase inhibitors that are derived from natural products. Okay, you saw all of those are synthetic, purely synthetic compound. We even mentioned their chemical synthesis. Okay, so what about the natural products? Sirolimus, or it's called rapamycin, it's a macrolide. It's a natural product used as an immunosuppressant during kidney transplant. But the more polar analogs, the temsirolimus and the everolimus, are analogs of sirolimus. These are more polar. These analogs have been approved for the treatment of certain types of tumor, where a kinase enzyme that is called mTOR or FRAP is excessively active. So if you use these agents in the types of tumor that have this enzyme excessively active, you will get better results, or let me say good results. The mTOR triggers a signal transduction process leading to cell growth, and so its inhibition serves to inhibit tumor development. And this is the whole idea. Okay, you need to inhibit tumor development and growth. This is the structures. For sirolimus, there is just a hydroxyl group here. And for temsirolimus, it's an ester. You see C double bond O, C methyl, CH2OH by 2. This is an ester. Instead of OH, it's going to be an ester. While in everolimus, this is just an ether. It's an O, CH2, CH2OH, ether with an alcohol at the end. The important thing with these agents, it's their mechanism of action. Okay, it has a unique mechanism of action. Okay, we saw in the previous slide that it inhibits the mTOR kinase enzyme, right? But it inhibits that enzyme in a special way. It has a special mechanism of action. And I think you like the mechanisms of actions because you are students of the College of Pharmacy. Well, without further ado, first, before it binds to the mTOR enzyme, these molecules have to bind to what's called FKBP12. Okay, it has this binding region that binds to this protein. After it binds to this protein, it binds to the mTOR enzyme by the TOR binding region, which is this one. Okay, so actually it brings those two proteins together. Okay, because it binds one protein at the side and the other protein on the other side. It brings those proteins together, and because these agents bring those proteins together, that results in the inhibition of the mTOR kinase. Okay, it resembles a sandwich. Okay, you know a sandwich, right? This resembles a sandwich, two proteins, and the agent is in the middle. It's just like a hamburger, where you have the hamburger in between two buns, right? This is the unique mechanism of action for these agents. And with that, I would like to thank you for your listening. And this is the last part. It's part 5 of our lecture. I hope that we can continue with the next lecture. And bye-bye.